Hello, welcome to this presentation on gene annotation of mycobacteriophages, ShaboiShabazz, and Aubs. My name is Danny.
My name is Smita.
And we are recording on behalf of our Phage Lab class at Virginia Commonwealth University.
Our initial goal was to isolate DNA from phages collected from soil samples. Using the host bacteria called Mycobacterium smegmatis, a non-pathogenic species having a rapid rate growth, we were able to isolate and purify the DNA from two Mycobacterium phages. ShaboiShabazz was isolated from soil collected directly from the VCU campus. Aubs was isolated from soil collected from a backyard in Northern Virginia. After obtaining results, it was found that both are Cypoviridae, meaning they have double-stranded DNA genomes, as well as non-contractile tails. These phages were purified and DNA was isolated for genome sequencing.
The phage ShaboiShabazz had a length of 41,901 BP. ShaboiShabazz had 61 open reading frames, 20 with functional annotation. It had 0 tRNA and a percentage of 66.6 guanine cytosine content. And characteristics were a 3 sticky overhang and 11 bases. The phage Aubs had a longer length of 58,937 BP. In Aubs, there were 107 open reading frames, 23 with functional annotation. There was 1 tRNA and a percentage of 61.4 guanine cytosine content. In characteristics included a 3 sticky overhang and 10 bases.
Once our genome was sequenced, we moved on to gene annotation. This consisted of 2 steps, position and functional annotation. Our first step to positional annotation was to use PECAN, the Phage Evidence Collection and Annotation Network, to view all of the predicted potential start positions that Glimmer and GeneMark have identified for each specific gene. Glimmer and GeneMark are programs that predict where the protein coding genes are located. So these tools go through the nucleotide sequence and mark every sequence that signals a start codon, AUG, ATG, and TTG, paired with a stop codon as a potential open reading frame. GeneMark places upticks for start codons on the coding potential map and downticks for the end codon. Glimmer identifies the start position TTG and lists it in the table of start positions, but output does not appear on a map. Using the predictions that Glimmer and GeneMark provide, we navigate the potential start positions through a series of criteria. Under the heading gap, we can see the number of nucleotides between the previous gene and the gene we are studying. A negative number in this column indicates an overlap. The best start position should have minimal gap or overlap scores. The ribosome binding site score consists of the spacer, z-score, and final score. The spacer represents the distance the ribosome binding site sits upstream of the start codon. The z-score represents how well the sequence is conserved. The final score is logarithmic, so the least negative score is the best. The best start position should have a spacer between 4 and 14 nucleotides, and the highest z-score and final score. The last criteria we can fulfill from PECAN is if the start position has the longest open reading frame. The start position with the longest open reading frame will have the longest sequence. Then, using NCBI BLAST-P, we can compare our choice to previously annotated phages in the GenBank database to see if other genomes are annotated in the same way. The different start positions PECAN provides have different protein sequences. So when these protein sequences are analyzed by BLAST-P, the best start position should have a majority of 1-to-1 matches. We use the GeneMark coding potential map accessed through PECAN to determine if the start position we have chosen covers the complete coding potential of the phage genome, which is the red line, and the host bacteria genome, which is the black line. If the start position does not cover the complete coding potential, it cuts off part of the sequence that could code for important amino acids. These images represented the positional annotation of ShaboiShabazz gene number 2. We chose the start position at 359 because it has the longest open reading frame and the best ribosome binding site scores. It also has many one-to-one matches on BLAST-P and covers the full coding potential on the map. Although this start position does not have an ideal score for gap overlap and was not predicted by Glimmer or GeneMark, we chose this start position because it has many one-to-one alignments in comparison to the 455 start, which has a 1:33 alignment.
To predict functional annotation, we use a combination of BLAST-P conserved domains database, HHPRED for comparison to proteins with determined structures, and TMHMM for transmembrane domains. Using BLAST-P, we compared our phage sequences with predetermined alignments in the conserved domains database and used hits with e-value less than 10 to the -5. HHPRED database's predictions were made by examining our phage sequence matches for hits with a probability over 90% and e-value less than 10 to the -7th power and a peer-reviewed publication associated with the match. Matches were also determined using TMHMM, which detected transmembrane domains in our phage sequence. According to the data shown by these three databases, we can annotate this gene as a large subunit of terminase. According to the peer-reviewed publication that goes along with the HHPRED database, we can further narrow this annotation down to large subunit terminase in the ATPase domain.
Each phage genome sequence was submitted to BLAST-N pairwise sequence alignment tool to identify the best sequence match in the GenBank database. In our collection, ShaboiShabazz's best BLAST-N match is Cetasite, and Tom Brady's best BLAST-N match is Crespo. ShaboiShabazz and Tom Brady are both cluster G1 phages, and this is a cluster that retains high sequence identity. In this table, we show ShaboiShabazz simply compared to Tom Brady because they're almost identical. Note that Tom Brady is 2 nucleotides longer than ShaboiShabazz, so they are unique sequences. Aubs' best BLAST-N match is Donkey Kong, and both of these phages are from cluster F1. Aubs and Donkey Kong have a 92% identity across their alignment. Note that these two F1 phages have no significant match to the cluster G1 phages, showing that phages from different clusters are unrelated at the nucleotide level.
Using the BLAST-N pairwise sequence alignment tool and comparing our collection's best matches to each other, we analyze a dot plot. This graphical tool gives a visual display of the similarities between the genome sequences, specifically each nucleotide being compared to the other. We found that ShaboiShabazz on the x-axis and its best match, phage Cetasite, on the y-axis, both of which are cluster G1 phages, have a straight solid line for the plot. This is due to its 100% identity. However, the second plot displaying Aubs on the x-axis and its best BLAST-N match, Donkey Kong, on the y-axis, has a more scattered line, both of which are in the F1 cluster. This is due to only a 92% identity between the two phages.
Famarator maps were examined for a genome-wide view of nucleotide and protein sequence similarities and differences. Shown here are the Famarator maps for each of the phages: Aubs, Donkey Kong, ShaboiShabazz, and Tom Brady. Some key notes when looking to analyze these maps. The colored shading represents levels of nucleotide similarity between the compared genomes. The colored gene blocks represent protein groups. The maps visually show us the comparisons in the length of genome as Aubs and Donkey Kong appear longer than ShaboiShabazz and Tom Brady. Aubs and Donkey Kong are a part of the F1 cluster, and ShaboiShabazz and Tom Brady are a part of the G1 cluster.
Combined, we have learned after the phage's discovery how to correctly sequence, annotate, and the fundamentals of these processes associated with our collection, specifically learning about the nucleotide sequence and proteins in comparison with other phages, various types of proteins and their functions in our phages, identifying coding potential, and much more. Through learning how to utilize multiple tools for our development of the analysis of our collection, we better understand their specialties, essentially their similarities with others in the database, and their differences that make them unique. Thank you for your time and patience. Thank you to the HHMIC program for allowing us to present our collection. We hope you enjoyed our presentation. Have a good one.